In eukaryotes, replication occurs in which phase of cell cycle? A. G1 phase, B. G2 phase, C. S phase, and D is M phase. So the right answer is answer C, that is the in S phase. So you can see here in S phase the DNA synthesis or the DNA replication is occur. The fragments of DNA are joined together by which of the following enzymes? A. Endonuclease, B. DNA polymerase, C. Primase, D. Ligase. So the right answer is D. Ligase. Ligase help in the joining of DNA fragments together. Association of histones H1 with nucleosome shows the occurrence of transcription, the occurrence of replication, exposed DNA double helix, the condensation of DNA into chromatin fiber. So the right answer is the condensation of DNA into chromatin fiber. Question 4. During replication, Okazaki fragments elongate. Leading strand towards the replication fork, lagging strand toward the replication fork, leading strand away from the replication fork, lagging strand away from the replication fork. So the right answer is answer D, lagging strand away from the replication fork. Okazaki fragment movement or the lagging strand movement is always opposite from the replication fork. Question 5. Which of the following enzymes separate the two strands of DNA during replication? A. Gyrase, B. Topoisomerase, C. Helicase, D. DNA polymerase. So the right answer is C, helicase. You can see here it's a leading strand, it's a lagging strand, and lagging strand synthesis always occur by these Okazaki fragments, and this is the replication fork movement and this is the leading strand movement, but the Okazaki fragment or the lagging strand movement always opposite from the replication fork. Termination of replication is triggered by DNA polymerase, helicase, SSB, and TAS protein. So the right answer is D, TAS protein. DNA polymerase synthesize DNA in 5' to 3' direction, DNA in 3' to 5' direction, mRNA in 3' to 5' direction, D, mRNA in 5' to 3' direction. So the right answer is answer A, option A, DNA in 5' to 3' direction. In some viruses, RNA serves as the storage of genetic material and DNA is synthesized from RNA by the enzyme known as DNA synthetase, DNA polymerase, reverse transcriptase, and DNA convertase. So the right answer is the answer C, reverse transcriptase. That's use RNA to form this DNA. Which of the following process does not occur in prokaryotes? Replication, transcription, translation, and splicing. So the right answer is D, splicing, because splicing is the process by which introns are removed, but in case of prokaryotes there are lack of introns, so splicing mechanism don't need for prokaryotes. In eukaryote, which of the following DNA polymerase is required for mitochondrial DNA replication? Pol alpha, pol beta, pol gamma, and pol delta. So the right answer is pol gamma, that's responsible for mitochondrial DNA replication. So here are some eukaryotic DNA polymerase, and DNA polymerase gamma helps in replication in mitochondrial DNA. Telomerase are the repetitive sequence of T and G that are present in the eukaryote to protect the random cleavage from nucleases. These telomeres are synthesized by the enzyme telomerase. Which of the following are the properties of enzyme telomerase? A. Telomerase is a reverse transcriptase enzyme. Telomerase consists of an RNA sequence that serves as a template. After completion of each cycle, telomerase translocate to 3' of DNA to synthesize repetitive sequence. D. All of the above. So the right answer is D, all of the above. It's a telomerase enzyme. These telomerase enzymes are reverse transcriptase enzymes, and it's have some RNA sequence, and these enzymes always bind to the 3' end of the DNA, translocate to 3' end of DNA. DNA polymerase 3 holoenzyme polymerase activity only, 3-5 endonuclease activity, 3-5 exonuclease activity and polymerase activity, and 5-3 exonuclease activity. So the right answer is C, 3-5 exonuclease activity and polymerase activity. So these are the prokaryotic polymerase, DNA polymerase 1, which have polymerase activity as well as 5-3 and 3-5 exonuclease activity. DNA polymerase 2 that have DNA repair ability and also 3-5 exonuclease activity, and DNA polymerase 3 that have polymerase activity as well as 3-5 exonuclease activity. So the right answer is C, 3-5 exonuclease activity and polymerase activity.